Lifespan. Chapter 1. Why we age. Most of us push thoughts of our mortality into the deep recesses of our minds. We try to avoid thinking about the ventilators, broken bones, surgeries, and hospital bills that have come to symbolize old age. But what if it didn't have to be this way? What if we could slow, or even reverse, aging so our final years could look like the ones before them? In Lifespan, David Sinclair reveals breakthrough findings on why we age and why we don't have to, including those from his own lab at Harvard Medical School. In the next chapters, you'll explore Sinclair's information theory of aging, that the key to stopping the aging process as we know it is in the packaging of our DNA. And you'll see how we can increase our health spans, the portions of our lives spent without disease or disability, thereby living not only longer but also better. To begin, what exactly is aging? Aging is scientifically understood as the deterioration of bodily functions necessary for survival and fertility. It's a concept we're all familiar with. Most of us understand that with age come changes in the color of hair, fur, or feathers and worsening eyesight, for instance. But the answer to why we age has been elusive. Various theories have been proposed over time. Early thinkers like Aristotle thought we aged and died for the good of the species, leaving room and resources for younger individuals, still capable of reproducing. This concept was widely accepted until the second half of the 20th century. Then, in the 1950s, rather than focusing on the good of the species, theories suggested that individuals aged once their genes were sufficiently passed onto the next generation. In humans, for example, bodies that were robust at age 18 and through prime reproductive years would begin declining after age 40. For various reasons, these theories of aging haven't satisfied scientists. Thus, the scientific community agreed on a new model, one suggesting that maybe there isn't a single cause of aging after all. Following this model, if there was a way to keep stem cells from tiring out, they could continue creating the cells needed to heal tissues and battle disease. And, if we killed off senescent cells, those that refuse to die and pile up in our tissues, we could stop them from disturbing other cells, keeping us healthier for longer. The other hallmarks could be individually addressed as well. Overall, researchers began to agree, if you address some of these hallmarks, you can slow down aging. If you address them all, you may not age at all. Sinclair takes things a step further. He explains why these hallmarks occur in the first place, and, in doing so, proposes a singular reason why we age, one based on the packaging of our DNA. You may be familiar with DNA as a thread-like double helix. Each human cell contains approximately 6 feet of DNA if stretched end-to-end in this form. Yet, the nucleus, the home of DNA, of a human cell, which contains the DNA, is only about 6 micrometers in diameter. Therefore, DNA must be carefully packaged into a compact structure. The epigenome does this, packaging our DNA so it fits in the nucleus of our cells. According to Sinclair, a loss of the information driving this packaging process is the single cause of aging. And protecting it is the key not only to eternal life but to eternal health. We'll explore this information theory of aging in more detail next. Chapter 2. The information theory of aging. Lifespan. As we learned, despite recent scientific focus on the multiple hallmarks of aging, Sinclair still believes that aging has a single cause. The cause? A loss of information. Information, essential for all life, comes in 2 forms: 1. Genetic information in the genome, and 2. Epigenetic information in the epigenome. Genetic information is digital, or made up of finite digits. In DNA, these digits are the nucleotides of adenine, C cytosine, T thymine, and G guarnine. Because DNA is digital, it is a reliable way to store and copy information. This information includes, for instance, the instructions for building proteins. But these proteins aren't made until the right genes are turned on. The epigenome turns on and off our genes in response to environmental cues such as nutrition, stress, and medication, and thus requires flexibility. That's why epigenetic information is analog. It's made up of infinite values, so that our genes can be activated to best respond to different and even novel situations. For instance, in a muscle cell, the epigenome turns on a set of genes that make muscle-specific proteins, while turning off those that are not of use to the muscle cell. Healthy epigenetic processes will also turn on genes that suppress tumor growth. Disruption of these processes, however, could turn off these genes, causing cancer. While many scientists believe that aging is caused by the erosion of this digital, genetic information, Sinclair disagrees. After all, consider cloning experiments. We can create young clones using DNA from old cells. That means most digital information remains intact with age. Sinclair's information theory of aging instead proposes that the loss of epigenetic information is what causes aging. To turn certain genes on and off, the epigenome wraps up segments of DNA, spooling up the genome just like you may spool up a garden hose. When the DNA is spooled up tightly, the genes in that region of the genome switch off. Those that are exposed, however, switch on. This packaging determines each cell's identity, and if the analog information guiding it erodes, the identity of our cells degrades too. Why does our epigenetic information degrade if our genetic information, for the most part, does not? Recall that epigenetic information is analog, not digital. It stores data as infinite, rather than discrete, values. This enables cells to respond flexibly but also makes the epigenome more susceptible to damage. So unlike its stable, digital counterpart, analog information degrades over time. And it can't be copied. As we and our epigenomes age, our cells start to lose their identities. For example, a nerve cell in an older person is no longer fully a nerve cell. As the epigenetic information guiding it degrades, it starts to become a senescent, old, zombie cell. This may cause forgetfulness. The degradation of our epigenome is also why our hair grays, our skin wrinkles, and our joints begin to ache, Sinclair says. Moreover, it's why each of the hallmarks of aging occurs. How can we preserve our epigenetic information? We'll explore this next. Chapter 3. Activating longevity and vitality. Within the epigenome, a certain kind of gene has been shown to extend the length and quality of life in many organisms. These genes are called longevity genes, and over 24 of them have been found in the human body. These genes create proteins that sit on specific areas of DNA and decide how to package it. The primary task of longevity proteins is keeping us healthy by maintaining the structure of the epigenome, as dictated by the environment within and outside of the cell. The proteins release compounds into the bloodstream that tell us to save energy in suboptimal conditions and grow quickly in favorable ones. But longevity proteins have a secondary role, a role that can distract them, repairing DNA damage. In the previous chapter, we learned that our genome remains largely intact as we age. But that doesn't mean DNA is immune to physical breakages. These breakages can form as a result of exposure to radiation and chemicals and during processes like DNA replication. Think of a scratched-up DVD, for example. It still holds the data needed to play a movie, even if it has external wear and tear. Similarly, a broken DNA strand still holds our genetic information, even when it's physically broken. If not repaired, breaks in our DNA can cause cells to die or form tumors. When longevity proteins sense broken DNA, they frantically move from where they should be to try and repair the damaged DNA. This works well when we are younger. But as we grow older, longevity proteins are tasked with repairs more often. And if they are faced with too much DNA damage, they can't keep up. They leave their typical responsibilities to address this damage, sometimes turning off genes that aren't supposed to be turned off. Sinclair hypothesizes that aging occurs when these longevity proteins don't go back to where they are supposed to be after fixing a break. Preserving our longevity genes and proteins is all about finding the right level of biological stress, says Sinclair. Longevity proteins are activated, turned on, in response to this stress, helping us survive longer in times of adversity. The right amount of biological stress slows cell growth and conserves energy, and still repairs DNA, all without causing lasting damage. According to Sinclair, that little bit of stress prompts the rest of the system to hunker down, to conserve, to survive a little longer. That's the start of longevity. Overworking them, making the longevity proteins run around after various DNA breakages, can cause lasting damage to the epigenome and lead to aging. Exposure to toxins such as cigarette smoke, pollution, and UV light, for example, can cause this strain. The right stressors will actually activate longevity and vitality. Next, you'll learn how you can activate these longevity genes with good stressors such as dieting, exercise, and cold exposure to ward off disease and aging. Chapter 4. Longevity Now. Research labs around the world are developing and studying anti-aging medical therapies and life-extending technologies. But even without access to those, we can engage our longevity proteins. We can control the behavior of our analog epigenetics by how we live our life right now. In the mid-2000s, writer Dan Buettner introduced the world to blue zones, regions in the world where people live longer than average. Since then, those seeking to apply lessons from longevity hotspots such as Okinawa, Japan, Nicaragua, Costa Rica, and Sardinia, Italy have focused on what blue zone residents eat. Although humans are all different enough that our diets should be at least subtly varied, we're also all similar enough that there should be some broad similarities. In general, we should be eating more vegetables, legumes, and whole grains, and less sugar, dairy products, and meat. Although meat contains all 9 of the essential amino acids, the organic compounds from which proteins are made, a diet high in meat is not ideal for a long and healthy life. Heavily animal-based diets can result in high cardiovascular mortality and cancer risk. Red meat, for example, contains a compound that the gut transforms into a chemical linked to heart disease. We can also obtain amino acids from plant-based protein sources, though plants usually deliver limited amounts per serving. From a vitality perspective, though, that's good news. A short supply of amino acids stresses the body out just enough to engage our longevity genes. This stress forces our bodies to hunker down, helping us survive longer. Our cells spend less energy dividing and more energy recycling damaged proteins. What we eat isn't the only important factor. How we eat also matters. After 25 years of researching aging, Sinclair recommends that we eat less. Studies have shown that specific families of longevity genes activate when on the razor's edge through calorie restriction, just enough food to function in healthy ways. Sinclair believes that a good target for this is 25% fewer calories than typically recommended, but studies have seen benefits in even a 12% calorie restriction. Caloric restriction tells our longevity genes to keep us alive during times of adversity by boosting cellular defenses, minimizing epigenetic change, and slowing down aging. In 1978, biologist Yasuo Kikuwa discovered that children on the blue zone island of Okinawa consumed 75% fewer calories than the children in mainland Japan. Adult Okinawans consumed about 20% fewer calories than those on the mainland. As a result, Okinawans' lifespans and healthspans were longer, with significantly less cerebrovascular disease, malignancy, and heart disease. Sinclair says that knowing this should not drive us to malnutrition or starvation, but instead fasting, allowing our bodies to exist in a state of want. Intermittent fasting, eating normal portions of food but with periodic episodes without meals, is one way to use hunger to engage our survival circuit. To trigger our longevity genes, we can also induce some biological stress through exercise. Inducing stress through exercise activates longevity genes that improve DNA repair, boost memory, and increase exercise endurance. These activated longevity genes instruct our bodies to grow longer and more protective telomeres, caps that shield DNA from damage. We don't even have to exercise for hours on end to extend our telomeres. A 2017 study funded by the US CDC showed that jogging 5 days a week for at least 30 minutes made telomeres appear nearly 10 years younger, i.e. longer. When exercising, just aim to raise both your heart and respiratory rate. Even better if you can do so outdoors in a t-shirt on a cold winter day. Or follow up with a cold shower. Exposing your body to cold temperatures also activates your longevity genes. Chapter 5. Future anti-aging pills. The more often a cause shows up on a death certificate, the more attention society gives to fighting it, says Sinclair. While age is sometimes considered an underlying factor at the end of many people's life, doctors never cite it as an immediate reason for death. But aging meets all criteria of being a disease, an illness or sickness characterized by specific signs or symptoms. So, Sinclair asks, why would we choose to focus on problems that impact small groups of people if we could address the problem that impacts everyone? Once we understand the regulators of aging and how to change them and recognize aging as a single disease, it will be remarkably easy to tackle. Many huge medical innovations have been simple. We stopped most surgical infections by washing our hands, and many diseases have been eliminated with vaccines. In this chapter, we'll explore the similarly simple but cutting-edge research currently in development to fight aging. One such line of research involves finding ways to kill off zombie cells. Recall that epigenetic breakdown can cause our cells to eventually lose their identities. These zombie cells secrete factors that accelerate cancer, inflammation, and turn neighboring cells into zombies. It's difficult to reverse aging in these senescent cells, so the best thing to do is to kill them off. Drugs called senolytics are in development to do just that, and they could rapidly rejuvenate us. The first human trials of senolytics were started in 2018 to treat arthritis and glaucoma, conditions in which zombie cells accumulate. Sinclair states, it will be a few more years before we know enough about the effects and safety of these drugs, but if they work, the potential is vast. In addition to senolytics, rapamycin, first discovered as an antifungal compound and an immune system suppressor, has been consistently successful at extending life. If you put 2,000 normal yeast cells into a culture, few will remain viable after 6 weeks. But if you feed those yeast cells rapamycin, in 6 weeks about half will still be healthy. Rapamycin also boosts energy and cellular repair by activating the longevity proteins we previously learned about. Similarly, metformin, a molecule used to treat type 2 diabetes, may be an effective longevity medicine by mimicking calorie restriction. Remember, caloric restriction can help slow aging by introducing the right level of biological stress. Metformin stresses our longevity proteins out just enough to inhibit cell growth, keeping our organs younger and healthier. In some places, such as Thailand, metformin is even available over the counter at every pharmacy, for just a few cents a pill. Elsewhere, even if you have prediabetes, it can be hard to find a prescription for it. Chapter 6. A world of longer, healthier lives. Lifespan. Taken separately, any of the pharmaceutical innovations mentioned in the previous chapter may provide extra years of healthy life. But, together, Sinclair believes they could provide decades of longer, healthier life. According to Sinclair, it's reasonable to expect most people alive today to live to 100 years. 120 is our known potential, one that many people could someday reach with the right technologies. And if epigenetic reprogramming continues to develop, 150 years might even be possible for people living on the planet with us right now. Ultimately, there is no biological limit, no law that says we must die at a certain age. With increased health and lifespans, Sinclair suggests we strive to die quickly and painlessly when we are ready, with counseling and rules guiding our decision. The question then becomes whether we can continue to develop the technologies to survive in the face of the consequences, challenges, and risks of population growth. Many environmental, social, political, and economic implications arise in a world where people live longer and healthier. We already see the effects of our extended lifespan in modern times. In November 2016, the late physicist Stephen Hawking predicted that humanity had less than 1,000 years left on our fragile planet. A few months later, he revised his estimate downward by 90%. Even if the current research only extends lifespans by a decade, the corresponding population growth could push our planet to breaking point. Environmental crises caused by our waste, effluent and carbon emissions and consumption, food and fossil fuels, for example, will be exacerbated. But Sinclair claims that population growth doesn't guarantee misery, disease, and starvation. Instead, it could bring about innovation. Think of London of the 1860s, which had a population of 3 million and was riddled with disease and poor sanitation. Today, London has 9 million residents and counting, and far less death, sickness, and to spare. The problem isn't how many people lived in the city but how they lived in the city. Sinclair suggests addressing consumption by furthering the current movement of dematerialization, in which physical items have been replaced with digital ones. Consider, for example, the pages and pages of documents that have been replaced by computer files. Additionally, in a world where people are healthier for longer, we would also have to change how we view and define work. Pensions and retirement plans would need to be re-evaluated as more people reach 65. Skillbaticals, subsidized time given to employees to acquire new skills, could also be implemented. Or mini-retirements for those who are secure in their careers, a year off to refresh by traveling, picking up a new hobby, or volunteering. Sinclair also recommends that, in a world with longer health and lifespans, age and economics no longer dictate medical treatments. Increased health spans and lifespans could increase wealth disparity. According to Sinclair, those with money should not be able to benefit from growing healthier, older, and richer, while others suffer through shorter, diseased lives. Sinclair also believes treatments shouldn't vary too much by age. Treatments keeping us healthy as we grow old should be offered to the young, and elders should have access to the treatments routinely used on the young. The main ideas, Chapter 6. There is no biological law saying that we must die at a certain age. The environmental, social, political, and economic implications that arise in a world where people live longer and healthier could be combated by innovation.